Basic unit of life. Organizational levels of organisms you take, there are 5 organizational levels. They are cell, tissue, organ, system, organism. Cell, it is the basic building unit of organisms, or else we can say that is the structural and functional unit of organisms. So tissue. Tissue is a group of cells with a common origin organized to perform specific function or functions. A number of cells together collectively having the same origin, same point of origin, they have been organized to perform specific function or function. Organs. Organ is a collection of tissues arranged together to perform specific functions. All these systems you take, okay, so these are organized to perform specific functions, and all the systems work interrelatedly together to form up an organism. Next we are going for the basic unit of life. Below that, another topic, history of the cell. So how these cells have been introduced. There was a person called Robert Hooke. He was a scientist, right? He was the first person to observe a thin section of a cork plant cell under the microscope. You can see the image of the person Robert Hooke as well as the microscope he has used in 1665 is what given on the diagram. So nowadays, what is the microscope we are using? It's completely advanced, right? So he was the first person to observe the cells of a thin section from a plant cell. When he was observing that, he could identify, he could recognize that there are some small rooms like chambers like structures. So these chambers are what he termed as cells. Okay, and this is how the word cell has been originated. And now there is another person. He is Antony van Leeuwenhoek. He is the first person to observe the cell structures of bacteria and euglena for the first time. You see, there are two different names with two different discoveries. Robert Hooke, he was the first person to observe the cells, right? When it comes for Antony van Leeuwenhoek, he was the first person to observe the cells of microorganisms. You see the difference? Commonly organisms, the first person was Robert Hooke, and microorganisms, the first person was Antony van Leeuwenhoek. Okay, and what he has observed under microscope is what given on the diagram in the image number 3, the cells in section of the cork. All right, so this is how, okay, the origin of cells have been placed. Regarding these cells, there were many people discovering about that, researching on that, and later they have formed up a cell theory. So this cell theory was formulated by 3 scientists: Matthias Leiden, Theodor Schwann, Rudolf Virchow. So these are the 3 people who have formulated, and there are 3 tenets of cell theory. What are they? The structural and the functional unit of life is cell. That means all the organisms you take, whatever the organisms you take, the structural and the functional unit is cell. And the second tenet is all organisms are made up of 1 or more cells. That means when we take the organisms, some organisms are there, they do have only 1 cell. In fact, the very first organism to be evolved on earth was also having only 1 cell. So those organisms we name as unicellular organisms. And if they do have more than 1 cells, many cells, more cells, they are termed as multicellular organisms. And we, being human beings belonging for animals as well as the plant, all of us belong for multicellular organisms. And the third tenet is new cells are formed from pre-existing cells. That is, from previously existing cells, due to the cell division, cells will be divided. Due to the cell division, they form up new cells. So that is what the third tenet here, new cells are formed from pre-existing cells. So accordingly, these are the tenets of cell theory. Again, I'm repeating with the structural and the functional unit of life organisms is the cell, or even we can say in this way too, the basic building unit of all organisms is cell. Both the ways are correct. Second tenet, all organisms are made up of 1 or more cells. New cells are formed from pre-existing cells. So we'll go to the next one, types of microscopes. When we come for the observations, we do have eyes to see the things, but there is a limit. We can't zoom in any stuffs, any objects, we can't zoom in as to observe by our naked eyes. So to clearly see the things which are very, very much smaller, we have to go for some instruments, equipments. And that is how the microscopies have been introduced. Accordingly, the very first microscope was a simple microscope. It was a hand lens. Hand lens in the sense, a convex lens. Okay, a convex lens has been fitted up with a handle, and that is what we use as a hand lens or a simple microscope. And thereafter light microscopes have been introduced, or compound microscopes. That is what usually we use in our school laboratories too. And remember the difference here: simple microscope consists of only one convex lens. Light microscope consists of two convex lenses with two different curvatures. They do have different focal lengths, right? Sometimes there will be two more additional two as to improve, as to advance magnifications. All right. So light microscope is used to observe rough structure of a cell, cytoplasm, nucleus, chloroplast, and vacuole could be observed. You see, all these rough structures we can observe under light microscope. Then why do we want to go for electron microscope? Now let's say the size of the cell part or any portion of the cell becomes very much smaller. How much smaller? These organelles are smaller than 0.25 micrometers. If it is so, we can't observe them under compound light microscope, then we have to go for electron microscope. So the fine structure of a cell, the mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies could be observed under electron microscope. When we come for electron microscope, there are two types of electron microscope. They are TEM, transmission electron microscope, SEM, scanning electron microscope. Features of microscope. So why do we want to actually use these microscopes? That is because of microscopes, they do have two important features. One, magnification. It is the degree to which the size of an image is larger than the image itself. That means, what is the size of the object? That size will be enlarged, zoomed by microscopes. So that is magnification, zooming, simply to say. Okay, so this magnification is there for microscopes, but in our eyes, the human eyes doesn't have magnification. The second point is resolution. It is the degree to which it is possible to distinguish between two objects that are very close. Microscopes, they do have a larger range of this resolution. So this magnification and resolution we are going to compare now. The magnification power of human eye is nil. We don't have magnification in human eyes. Magnification power of light microscope is 1500 times to 2000 times. That means one object, one smaller object could be enlarged up to 2000 times. All right, this ability we don't have in our eyes. And when it comes to electron microscope, which are very much advanced, up to 500,000 times, up to 500,000 times we can get a magnification. So these are very much important when we are observing the viruses and all smaller microorganisms when we are observing, and smaller cell structures when we are observing. Finally, electron microscopes are important since they are giving up to 500,000 enlargements. And what is the resolution power? In our eyes, we do have a resolution power up to 0.1 millimeter. That is, if there are two objects which are closer to each other at a distance of 0.1 millimeter, we can see those two objects as two. We can distinguish them separately. But if the gap is less than 0.1 millimeter, if the gap is less than 0.1 millimeter, we can't distinguish them as two. It seems to be like one. So that we have to go for light microscope, and light microscope gives you a resolution power. The minimum gap between them should be 0.2 micrometer. So 0.2 into 10 to the power minus 6 meters, or 0.2 micrometer, is the light microscope resolution. And then electron microscope you take, even the range is bigger than that. That is up to 0.5 nanometer. Nanometer means 1 billionth. Micrometer is 1 millionth, right? Nanometer is 1 billionth. Even for that level, we can observe the things separately. So that is the resolution power of electron microscope. Cellular organization. We said now, based on how many cells are present in organisms, we divide them into two. What are the two? Unicellular organisms and multicellular organisms. Unicellular organisms, they are the organisms composed of only one cell, single cell, just like bacteria, okay, protozoas, right, amoeba, paramecium, euglena. All these are some examples for unicellular organisms. And multicellular organisms, animals and plants belong to multicellular organisms. Only a few examples I'm giving you. So who are the multicellular organisms? The organisms composed of many cells. The organisms composed of many cells. Fine. Cells perform different functions in the body. So whatever the cell you take, okay, these cells combine together to form tissues, we said, you could remember. And the tissues, we said, they do have specific functions. So accordingly, cells are contained in tissues, hence they do have different functions in our body. Some examples: the transportation of oxygen is done by red blood cells. Transportation of oxygen transporting to outer body is done only by red blood cells. It cannot be done by white blood cells, or it cannot be done by nerve cells. So they are specifically functioned. So you see now, neuron is adapted for the transmission of impulses, and that cannot be done by red blood cells. You can understand now, right? So all the cells tissues have been originated specifically for certain functions. Now the cells differ from one another from their shape, size and function. So when we take their shape, the sizes, the functions, or whatever the colors even, they are different from one another, and that is how we can identify, recognize them. We are going to observe certain cells now. You see the first one, microscopic observation of cheek cells. Using a finger or a nail spoon, okay, or whatever, what do you do now? You take some cells from the inner part of your buccal cavity, mouth, right, from the cheek. And then what do? You have to keep it over a glass slide. You have to apply a few drops of water, not wash it out with water, a drop of water as to fix it, and cover it up with a cover slip. And you have to make sure that no any air particles or any dusty particles enter there. Thereafter, what do you do? And sometimes, you know, we are using some strains too. And anyhow, at the end, we observe under microscope. When we are observing under microscope, there are some techniques you have almost learned in lower grades, all right? So you have to adjust with all the parts, and then you have to make sure you are starting it from the lower magnification, and furthermore, until you observe them finally, you can increase your magnification. So anyhow, when you observe that, you can see the second image given, a violet, a light purple colored cells are what you can see. This is how you can observe under microscope, the light microscope. And what is the type of cell is this? Human cheek cells. And the second example I'm going to take, the microscopic observation of epidermal cells of betel leaf. The betel leaf you take, this is also the same way. When you observe that under microscope, okay, this is how you can recognize the parts. You can see some small reddish colored dots like structures. They are what we term as, the portion is what we term as guard cells. Epidermal cells, that is the upper layer or lower layer. Upper layer we say upper epidermal, lower layer we say lower epidermal, right? Whatever the epidermal cells you observe under microscope, you can observe some guard cells. So these guard cells are termed there, and they do have epithelial cells too, and they are the largest cells here. So you can see now this cell structure is completely different from human cheek cells. So the same way, if you are going to do this for onion cells, onion tissue cells, we have to peel off some onion tissues. The same way we have to apply them soak in water, and then we have to take it over the glass slide. We cover up by a cover slide after applying a drop of water, and we observe under microscope. Those images are given. You can observe the structure of the cell under microscope of this onion tissue. Now we go for the organelles and typical cell structures. Two topics here. What is an organelle? You could remember these words. You have heard of these: cell wall, mitochondria, chloroplast, vacuole. So like this, certain words you have heard of. And these are what we term as organelles. And these are the small, tiny structures. Small, tiny structures present within the cells. Each of them, they do have different functions. So how do we write the organelle? The small structures present within the cell to perform different functions are known as organelles. What is a typical cell? Typical cell structure means that is the cell imaginary drawn including all the possible organelles, and there is no organism having all. Most of the organelles are present, but anyhow one or two will be missing on. So accordingly, typical cell, the cell prepared by including all the organelles, is known as typical cell. So in the living world, such cells do not exist, but cells with a certain number of organelles can be found in living organisms. You can see two rough structures given here. The upper one, these diagrams are given for you to identify, right? The upper one is the animal cell, and the lower one is the plant cell, a rough structure. So when you observe these two images finely, there is a difference. What's the difference here? You can see the upper animal cell does have only one outer layer. Can you observe that? Only one outer layer is there, and that is the cell membrane or plasma membrane we need. Okay, and observe the second image there. Can you see each cell you take, not only one outer layer, there is another two. two layers are there. The outermost layer in the second image is the cell wall, and the inner layer is the cell membrane. Accordingly, you can see here, animal cells do not have cell wall. Plant cells, they do have the cell wall. Thereafter, the cell membrane is present. I'm going to take now both the cells, typical plant cell and typical animal cell. Organelles and typical cell structures. The first one is the cell wall. When we take the cell wall is present only in plant cells. Cell wall is present only in plant cells, not in animal cells. This cell wall is non-living and fully permeable. It allows everything to exchange. What is the meaning of the word permeable? The penetration through the layer is possible for every part. Okay, and it is a non-living layer too. So the outermost covering of the plant is the cell wall, we said, and it is a dead structure. So non-living means it is a dead structure. What is the reason? This cell wall is made up of cellulose, and they are fibers. Hemicellulose and pectin are also present. You can see the diagram given here. This is a rough diagram for cell wall. What are the functions here? Maintain the shape of the cell. So because of this function, it can maintain the shape. The shape will not be changed, and it supplies support and protection to the cell because they do have cellulose fibers. So these are the three functions of cell wall. And the next one is plasma membrane or cell membrane, we say. Now, when it comes to this plasma membrane, that is the second layer after the cell wall, and that is the first layer in animal cells. The difference is plant cells, they do have cell wall and then cell membrane. But animal cells, since they don't have the cell wall, their outermost layer is the plasma or cell membrane. Plasma membrane is present interior to the cell wall of plant cells as the boundary of the animal cells, the outermost layer of the animal cells. It is made up of phospholipids and proteins. So what it says, cell wall, earlier we said, that is made up of cellulose. And the cell membrane or the plasma membrane you take, that contains phospholipids and proteins. Phospholipids, they belong for lipids. Proteins, okay, there is a type of protein here, we name it as globular protein. Phospholipids and globular proteins are what forming up the plasma cell membrane. Additionally, there will be cholesterol too. Okay, and it is a light semi-permeable membrane as well as a selective permeable membrane. So this is a living layer. Okay, this is not non-living. This is a living and semi-permeable. Cell wall, we said it is fully permeable, and plasma membrane semi-permeable. Semi-permeable refers to it can penetrate, it allows the penetration of only selected particles. Everyone can't go through. Okay, so that is what we say, semi-permeable or selective permeable. And what are the functions of them? Enclose the cell. Okay, they will be covering up the cell, especially in animal cells. Okay, allow entry of water, ions, some molecules, and thereby control the entry and exit of materials into and out of the cell. So we said now semi-permeable membrane. So what are the selected particles allowed to move through? Water, ions, some sort of molecules. So it controls the entry and exit of materials, being a semi-permeable membrane. So these are the functions. So there are two diagrams given here. The first diagram is of the animal cell, and the second diagram, it shows you the plasma membrane. Okay, so you can see upper and lower layers. Between that, small or long vertical layers are there. So they are the phospholipids, and round shaped, spherical shaped cells are there. They are the globular protein. So that is an additional diagram for you. Okay, so that is regarding the cell membrane. And now we come for cytoplasm. Cytoplasm. The gelatinous liquid part of the cell, excluding organelles, is known as the cytoplasm. So cytoplasm is the entire area inside the cell where it supplies the space for the cell organelles. That is something like I gave you an example, right? Like a plum pudding. So in a plum pudding, when the plums are taken, they are like the organelles. Okay, and the entire free space of the pudding, that is like the cytoplasm here. So cytoplasm gives the space for these all organelles. Inorganic and organic substances are present in it. So what are present in cytoplasm? Organic, inorganic substances are present here. The structures submerged in the cytoplasm are named as organelles. So we said you're right. Organelles are what the cell particles emerged in cytoplasm. And some organelles are surrounded by cell membranes. So when it comes to some sort of organelles like mitochondria, endoplasmic reticulum, Golgi complex, and nucleus, okay, as well as chloroplast. So they do have a specially designed cell membrane around themselves. That means when it comes to cell membrane, that is entirely surrounding the plant or animal cell. Okay, so these suborganelles that you take, those organelles also will be having a cell membrane. Sometimes one monocell membrane, sometimes bimembranes, two membranes. Okay, so anyhow, what you have to remember is mitochondria, nucleus, endoplasmic reticulum, Golgi complex, Golgi bodies are some examples for those having cell membrane surrounding themselves. And what are the functions here? You see, maintain a shape to the cell. So cytoplasm also will be maintaining the shape to the cell because cytoplasm is the interior part. If the interior part is not present, cytoplasm is not present, the cell could be squeezed. So this helps you at its presence to maintain the shape of the cell. Bare cell organelles, all the organelles are bearing by embedding in cytoplasm, carry out different metabolic processes. So different types of metabolic chemical activities are carried out inside the cytoplasm. And the next one is the nucleus. Nucleus, when we take, that is a living part, living layer, and this is what controls all metabolic activities, all cellular activities in a cell. Okay, either maybe animal cell or plant cell, or all the cells you take, all the living cells you take, they do have a nucleus. I said they control cellular activities. For these cellular activities, different words we can give: biochemical activities, biological activities, okay, cellular activities, metabolic activities. So these are the terms used for the major function of nucleus. So you see there, nucleus, which is a living organelle, is the main site that controls all metabolic cellular activities consisting of a nuclear membrane. So nucleus also does have a membrane, an outer layer that we term as the nuclear membrane or nuclear envelope. Two words here, nuclear membrane or nuclear envelope, which encloses the network of chromosomes, nucleolus, and nucleus. Actually, these are the particles present inside the nucleus. So inside the nucleus, you could remember we have named them in the typical plant animal cells too. Inside the nucleus, there is another spherical part that is the nucleolus. And there are some fiber-like structures. They are the chromosomes, and what it contains inside the nucleus, the liquid form, that is the nuclear sac. Okay, so these are the parts of nucleus. Surrounding the nucleus, there is a tubular structure present, and that is what endoplasmic reticulum we said. Okay, we have named them. You could remember the diagram. So endoplasmic reticulum, when it is combining with this nuclear membrane covering up the nucleus, is not a continuous layer. It does have spaces between it. Small spaces are present here and there between the layer. And these spaces, these holes are what we term as nuclear pores. Nuclear pores. So accordingly, nucleus, when we are naming separately, there are certain factors, words you should remember. That is, the outer layer of nucleus is nuclear membrane or nuclear envelope. Okay, and between the nuclear envelope, the spaces, holes are what we term as nuclear pores. And inside the nucleus, there is another spherical part, that is nucleolus, fiber-like structures, chromosomes, and the inner liquid stuff that are nuclear sac. So these are the parts of nucleus. One or two nucleolus and the chromatin body are present inside the nucleus. So what are present inside the nucleus? Maybe one or two nucleus and chromatin bodies would be present there. And during cell division, the chromatin body converts into chromosomes. You see now, cell division. This cell division is what we have to learn later. During the division of cells, dividing of the cells, chromatin body will be converted into chromosomes, and these are the major topics of nucleus. Then what are the functions? You see there. Nucleus, it controls all metabolic cellular activities. And nucleolus, it stores the necessary proteins in the nucleus. So what is the protein? What are the types of protein needed for these functions? That will be stored in nucleolus. Chromosomes, storage of genetic material and transfer inherited characteristics from generation to generation. We say, right, generation to generation, there are certain characteristics transmitted. So these will be stored in chromosomes. Okay, DNA is what the major part. So these particles will be stored inside chromosome. And do you know,
The number of chromosomes is specific to a species. Species to species you take, they do have a specific number, a unique number of chromosomes. When we come for humans, we do have 46 chromosomes, or we say 23 pairs of chromosomes, because chromosomes will be paired with one another. Okay, so single chromosomes 46, paired chromosomes 23. And in a frog there are 26 chromosomes, and in paddy plant 24 chromosomes. You can see they are different to each other. There are few examples, and this is about nucleus. Mitochondria. So mitochondrion you take, this is termed as specifically the powerhouse. So when it comes to a cell, mitochondrion is the place to produce energy. So that energy is what will be used for the entire process of cells. Okay. So this mitochondrion is termed as the powerhouse of the cell, since it produces energy by aerobic respiration. Aerobic respiration refers to at the presence of oxygen, it produces energy. That's correct, right? We inhale oxygen, right? This oxygen is what will be supplied through the blood circulatory system to our cells, tissues. And that is the place where the energy will be produced. That is done by mitochondria. Accordingly, what is its structure? It is an oval or rod-shaped bi-membrane bounded organelle. Can you remember we said earlier there are some organelles, they do have monomembrane, one membrane, some bimembrane, two membranes. So this is an organelle having bimembrane. There are two membranes. You can see in the diagram also. There are two layers surrounding the mitochondria. Aerobic respiratory reactions take place within the mitochondria to release energy. We said you're right, aerobic respiration. So it is known as the powerhouse of the cell. The energy produced within the mitochondria is used for the metabolic activities of the cell. So all chemical biological activities, what is the energy used? That is the energy produced in mitochondria. So accordingly, the only major function of mitochondria is production of energy by aerobic respiration. So the next one is chloroplast. Chloroplast you take, that is present only in plant cells, that is not present in animal cells. And this chloroplast contain a green color pigment called chlorophyll. Remember, inside the chloroplast that it contains chlorophyll, green pigments. Because of this only the plants get a green color. So it is mostly oval shaped membrane bounded organelle. So here too you can see this chloroplast also consisting of a membrane, and it contains chlorophyll pigments which give green color. What is its function? Photosynthesis. When you are learning about photosynthesis, you could remember simply that is the process of producing food. Okay, so how does it produce food? It needs four factors. What are the four factors? Carbon dioxide, water, sunlight, and chlorophyll. Carbon dioxide, water, sunlight, and chlorophyll. And carbon dioxide, water, sunlight, they are the external factors needed for photosynthesis, and chlorophyll is the only internal factor needed for photosynthesis, and this chlorophyll is present in chloroplast. So accordingly, the function is photosynthesis. And the next one, ribosomes. They are tiny small organelles without a membrane. So ribosomes you take, there are dot-like structures within the plant and animal cells, right? So they do have tiny structure and they don't have a membrane. Okay, they are membraneless organelles. They are made up of a large subunit and a small subunit. They do have two units actually. It is something like two spherical shapes or two circles you are keeping one over another. One is small, the other one is large. Such a structure it does have. Okay, one is a large subunit and the other one is a small subunit. They can be found freely in the cytoplasm or attached to endoplasmic reticulum. So where they can be found? Freely in the cytoplasm or attached to the endoplasmic reticulum. What is the function of ribosomes? Protein synthesis. They do produce proteins. Endoplasmic reticulum. Endoplasmic reticulum is a complex tubular system connected with outer nuclear membrane which performs the function of transport and is surrounded by a membrane. This transportation will be done from the cell out and out to the cell inside the cell structures. Okay, mainly this is connected to nucleus. It is an intermembranous network made up of flat or tubular sacs within the cytoplasm. So inside the cytoplasm that is present, and this endoplasmic reticulum, intermembranous we say. Why? Inside the membranes they are found. Okay, and there are two types of endoplasmic reticulum: rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum means they are the reticulums with ribosomes attached to the membrane, with ribosomes, and smooth endoplasmic reticulum without ribosomes. So this is how you term rough endoplasmic and smooth endoplasmic reticulum. And what are the functions? Separately, we are giving the functions for them. Rough endoplasmic reticulum, RER, that transports proteins within the cell. Okay, can you remember? You can combine certain factors here. Ribosomes produce protein. And where are these ribosomes? They will be present on rough endoplasmic reticulum, and that produced proteins will be transported by rough endoplasmic reticulum. That is a transporting pathway. And smooth endoplasmic reticulum you take, that will be synthesizing lipids and steroids. Lipids and steroids will be produced by smooth endoplasmic reticulum. And remember, steroids are also lipids, but they are hormones. So we are giving a specific name, steroid hormones, and to transport them within the cell. So producing lipids and steroid hormones and will be transported within the cell, that is done by smooth endoplasmic reticulum. So you can see the diagram given here for rough and smooth endoplasmic reticulums. And the next one is Golgi complex. Living and tubular organelle where the membrane bounded sacs stuck on top of the other with associated secretory vesicles are collectively known as Golgi complex. Look at the diagram given here. Tubular structures which are living, they have been stacked one over another. They have been connected to one over another. You can understand with the diagram there. And they do have some secretory vesicles to secrete out certain secretory substances, right? And this collection is what we term as Golgi body or complex. Why it is a complex form? It's it's combination form, right? What are the functions of them? Production of secretory substances. We said they do have secretory vesicles. So they will be producing secretory substances, some chemical substance. Storage and packaging. Storage and packaging. So all these particles will be stored and packaged. That is something like, that is the storage, where the storage, packaging, arrangement, everything would be done in this Golgi complex. And secretion, transport for cellular activities. So all these secretory substances will be transported to the cellular activities too. So these are the functions of Golgi complex. And the next one is vacuole. And this vacuole also is present only in plant cells, and a large central vacuole is present in plant cells. You can see the diagram here. A large central vacuole is present in the plant cell. But when it comes to animal cells, they don't have a permanent vacuole. Sometimes and smaller temporarily vacuoles may be present in animal cells. Okay, but that is not permanent. So you see there, it is a fluid filled a large non-living organelle found in plant cells. Okay, vacuole is non-living and it is filled up with liquids. And these liquids containing or the substances filled up within the vacuole is what we term as the cell sap. And the surrounding layer, can you see there is a layer out? This layer surrounding the vacuole, we name it as the tonoplast. So accordingly, sometimes small or no vacuoles in animal cells and contractile vacuoles can be found in unicellular organisms. So when it comes to unicellular organisms, one cell containing organisms, there may be contractile vacuoles present. It is surrounded by a membrane known as tonoplast. So here too one membrane is present, that is tonoplast. The fluid contained in it is known as the cell sap, and this cell sap contains many pigments, ions, water, sugar. And the functions of this vacuole are maintenance of water balance. So since it contains water, it stores water, it maintains the water balance, and it gives a support to why. It is something like, just imagine a balloon is filled up with water. A water-filled balloon becomes turgid, hard, tough. Same way, since this vacuole is filled up with water, it gives support, it becomes hard. Provision of color to the cell by the pigments within it, and also the color to the cell will be given by these pigments present inside the vacuole. So these are all about the cell organelles present maybe in plant cell or animal cell. And let me compare the differences between the animal cells and plant cells of what we have learned now. Cell wall is absent, cell wall is present. That's correct, right? Animal cell wall is absent, plant cell wall is present. And large content of it contains cytoplasm. That's correct. Animal cell, a large portion is containing with cytoplasm, but plant cells, when you take, it is pushed towards the periphery. Okay. Periphery means to a corner, cytoplasm is pushed because it contains a large central vacuole. Animal cell, a large vacuole is absent, but sometimes few small vacuoles temporarily may present. But plant cell, a large central vacuole or few vacuoles may be present. Okay, there may be a large central vacuole or few vacuoles may be present. And chloroplast is absent in animal cell, but in plant cells, chloroplast is present. So these are the differences between animal and plant cells.
To get notified about our next videos, subscribe to our YouTube channel Science with Anas Mohideen.